Hello, everyone. Thank you for coming. My name is Neil Krishna, and I'm from Boston, Massachusetts. I'm a sophomore in the special program in medicine, double majoring in physiology, neurobiology, and English. And this past summer, I worked in Dr. Joseph Laturco's lab on ependymoma. So today I'm going to be talking to you all about characterizing the molecular and genetic landscape of a brain tumor called supratentorial ependymoma. So a little bit of background. What is supratentorial ependymoma, or really just what is ependymoma? So ependymomas are glial cell tumors that form from primitive ependymal cells that line the inside of your central nervous system. So they form 6 to 10% of intracranial tumors in children, and actually, ependymomas are the third most common pediatric brain tumor. But nonetheless, ependymomas are divided into two subcategories, infratentorial and supratentorial. Infra meaning below the tentorium layer in the middle of your brain, and supra is above. I'll be focusing on the latter. So really little is known about supratentorial ependymoma in general. Nobody knows how it's caused, how it forms, and what effects it has on the body, except for a really poor prognosis. So I'm going to be looking at how we can find targeted therapies to combat this kind of cancer. So a little bit about the formation of supratentorial ependymoma, which is also called SE or STEPN. And there's a protein, it's a fusion protein caused by a chromothripsis between two different chromosomes. And this fusion protein is called the C11orf95 relay. It's a mouthful, it's just two different parts, the C11orf95 part and the relay part. And we know a little bit about the relay part, but we don't know much about the C11orf95 part. So one of the early parts of my project was trying to figure out which part of this chromosome has a hand in causing the formation of this tumor. So what I did was I mutated a single base pair in the C11orf95 part, a histidine to an alanine, and what we found is that the protein no longer caused the formation of tumors, which tells us that there is something to do in this part of the protein that causes ependymoma. And previous studies, and especially our lab, have shown that the relay domain of this fusion protein has a hand in tumorigenesis through something called the NF-kappa B signaling pathway, but that alone is not significant to cause tumorigenesis by itself. So here we see a little bit of that pathway, a snapshot. It's a common inflammation pathway, but by itself, it's not enough to cause this tumor. So what else is there? Well, so one of the big and major processes we can use to help figure this out is a tool called RNA sequencing. RNA sequencing comes in two varieties, bulk and single cell, where we look at a population of cells and the individual cells that make them up, and we see what genes are overexpressed, underexpressed, and how they're expressed. So my project started off with a human astrocyte transfection. The reason I used human astrocytes is because they're closest in form to the types of cells that ependymoma originates from in your central nervous system. I also did this experiment with other types of cells that I'm not going to show here. So I transfected the human astrocyte cells with two constructs. The first is the fusion protein, which we know causes tumors, C11orf95 relay. And the second is the fusion protein that's mutated that we know does not cause tumors, which is the C11, but with the histidine mutation. And then, after 48 hours, I harvested these cells, lysed them, prepared them for something called quality control, and prepared isolates to send to a company for RNA sequencing. A few weeks later, they sent me back my RNA sequencing. I did a bulk RNA sequencing, and now I have a huge mass of about 20,000 genes, which I had to sort through, filter in every way possible. So then began the computational bioinformatics part of my project, where I looked at these 20,000 genes and ran my own analysis in R called a DESeq2 pipeline, which I customized to suit my needs. And then we got data. So the goal, just to go over this one more time, is to look at what the function of the C11orf95 fusion protein plays in the formation of supratentorial ependymoma tumorigenesis, and what genes are implicated in that process. So this beautiful cloud plot is one of the initial slides of data I have, which show straight from the bulk RNA sequencing data which genes are upregulated and which genes are downregulated. So I applied a gradient filter on this, which put a gray density layer on all of the most dense cells. So everything above that regression line is an upregulated gene, and everything below that line is a downregulated gene. So this is already interesting. We can see that in the tumors, compared to the normal non-mutated tumors, non-mutated cells without tumors, there are a significant portion of upregulated genes. So you take all those upregulated genes and you run what's called a KEGG analysis on them, which looks at all the pathways that each of those genes is involved in, and you get this massive pathways. It's cool. There are a lot of names jumping out of us, like pathways in cancer or PI3 AKT signaling, but this is a lot of genes. This is about 2,000 genes, and we can't target every single one of them. So I had to apply a second layer of filtering. And very coincidentally, at the time I was doing this project, in June or July, a paper was published by Cancer Cell, and that was looking at single-cell RNA sequencing in supratentorial ependymoma versus bulk RNA sequencing, which I did. Now, single-cell RNA sequencing looks at a single cell, and they had 9 or 10 subtypes of tumor cells that they looked at. So here was the title of that paper, and what that means is they just looked at what genes are upregulated and downregulated in single-cell supratentorial ependymoma. And here are the subtypes of tumor cells that they used. These are just different subtypes of tumor cells. And what I did was I took my list of upregulated genes of about 2010, and I took their list of upregulated genes, which I consolidated into one big list of about 1,011 genes. And I crossed the two. And what it gave me was this list of about 155 common genes. So these are genes that my data says is upregulated and their data says is upregulated in bulk RNA sequencing and in single-cell RNA sequencing. So these are the most important upregulated genes, essentially. So then I reran the pathway analysis, the KEGG analysis, to look at how these genes are influencing the pathways in tumorigenesis. And what I found was that it's a lot simpler. There's one that stands out in red over here, the MAP kinase signaling pathway. And then we see the PI3 AKT signaling pathway is pretty large, as well as RAP1 and RAS. Those four actually are very interconnected pathways in their own right. They all form a huge part of signaling intracellularly, and they're all very heavily implicated in some other types of cancer. So after that, I'm sure all of you are wondering, what do these pathways mean? So here's a diagram which shows generally what each pathway does. For example, we see neuronal system development or cell differentiation, protein phosphorylation. And what's interesting about this is that it all makes sense. These are the things we would expect to see in tumor cells that are different from normal cells. For example, we would expect to see an increased cell cycle. They're cancerous, or increased signaling. There's all sorts of inflammation response against these tumor cells. So this was a little bit of a validation of our data, that first of all, our bulk RNA sequencing was solid data, and second of all, that it was producing usable information. So after that, I was curious on how the pathways looked per cell, per tumor subtype. So that is, what the probability is that given a cell with a particular gene, it is in a particular tumor cell subtype. So that's a little bit confusing, but look at this graph. We can see that if you have any particular single cell with an upregulated gene, the likelihood that it'll fall into one of these categories. And as you can see, 3 of these categories really stand out. These are also the 3 that we would expect to stand out, because over here on the far right, STYAP1, that's an alternative to Relay. The C11orf95 Relay fusion protein we have, YAP1 is the other theory. And we're glad that's not upregulated, because that lends credence to the idea that C11orf95 Relay fusion protein is playing a large role in tumorigenesis. After this, I looked at the quantitative breakdown of the pathways that are upregulated in this cancer. This is, like I said, a quantitative version of the colorful KEGG analysis I showed you on the last slide. So again, MAPK signaling pathway is heavily upregulated at about 20% of all upregulated genes, whereas these other pathways, which are all closely related, are also upregulated, but to a lesser degree. So from this data, I looked at each pathway individually, and I consolidated a list of cells. Remember, we started with 155, and without overlaps, it actually comes down to only 53 genes that are heavily upregulated and are in these pathways. In the MAPK signaling pathway, which is kind of a more broad pathway than the other related pathways, I located these 11 genes, and of them, 1, 2, 3, 4, 5, 6, 7 of them are viable gene targets. The way we determine if a gene is a viable gene target is by looking at what agonists, inhibitors, and antibodies exist for that particular gene. That is, can we target them in the lab? Is there enough science on them to target them? And of this pathway, with the genes that I identified, 7 of them are viable targets. Also, every one of these genes is interesting in some way or another. And if none of these gene targets work, we have 53 minus 7 left. So with that list of genes, the biggest thing is to replicate these tests in vitro. That means we take a population of human astrocytes, we treat against these genes, meaning we inhibit them or we overexpress them, and we see what happens to our tumor cells. So just to go over what I did, I took a two-pronged filtering approach to a huge set of 20,000 genes, and from them I located 7 target genes that we will be able to look at in vitro for the capability of inhibiting tumorigenesis in supratentorial ependymoma. So after we target these genes and pathways, the MAP kinase pathway especially, in vitro in a population of cells, with the genes that proved as the best targets, we moved to in vivo testing in a mouse model. We do the same thing. We knock out using CRISPR these genes, and then we look at if this prevents tumorigenicity in mice. Next, we also look at overactivating MAPK and RELA in vivo and in vitro, because these two pathways are so closely linked together, and they seem to play such a large part in tumorigenicity that doesn't happen in the wild type mutants without the tumor. So what would happen if we overregulate both these pathways? Would it inhibit tumorigenesis completely? Hopefully. So I'd like to give a big thank you to everyone who helped me in this project. Dr. Joseph Loturco, my PI, and a wonderful person to work with. He's the professor and department head of P&B, and I'm in his lab, and I hope to continue working on this project for many years. I'd also like to give a thank you to Dr. Vin Moscardelli, again, another wonderful person to work with. He's the director of the Holster Scholars Program. And of course, Mr. and Mrs. Robert and Carlotta Holster, who, without their insight, none of this would be possible. And my fellow Holster Scholars, friends, peers, and of course, my family, who helped me get through all of this remotely. Thank you. Are there any questions?